Hello and welcome to this tech talk about CRISPR. I'm Simon Carlton and I'm Anitox Technical Director for our EMA region, and I'd like to explain some recent developments in the world of salmonella mapping. Let's start by looking at human cases in Europe. The most recent report shows total cases are static, but salmonella infections acquired in the EU are increasing, as is the total number of outbreaks. So this is bound to remain under government and public scrutiny. How do raw materials and feed get contaminated? Well, animals in fields or flies, insects such as beetles, people spread in warehouses and mills, etc., and of course birds. Any site where raw materials are stored outside, even for a short time, provide a great food source for local and migratory birds, as you can see here. And we aren't just talking about salmonella here. Also Clostridia, avian influenza, African swine fever, PEDV, PRRS, E. coli and others have all been shown to survive in feed. Protecting feed should be a very important part of any biosecurity program for animals. Tracking where salmonella contamination comes from is fraught with difficulties. So let's have a look at some of the challenges. Salmonella in feed and ingredients are not uniformly distributed. If 100 salmonella are added to a ton of feed and evenly distributed, the odds of finding salmonella in a single 25-gram sample are 1 in 400. This, allied to the uneven distribution, gives us uncertainty in detection. How samples are taken and then analysed can also have an impact. Sampling error can include not getting enough or sufficiently representative samples. Subsample error can include not grinding the sample to allow good homogenizing of the sample to get good representation. We now know far more about methodology for recovering salmonella than when this study was done. So analysis error is likely to be more than 2% due to media bias and isolate selection bias. To give you an example, here we see salmonella isolates on a petri dish, and there are plenty of isolates. Typically, only one isolate will be picked to send off for serotyping. Some may send off two, but when each isolate costs around €70, that's unlikely to happen too often. The problem is there may be more than one serotype growing on the dish. The one or two picked are more than likely to be from the most predominant serotype. That is the serotype that has recovered quickest from the stress state it was in due to being in dry raw materials or feed. But there could be other serotypes here which get missed, ones which are present but do not predominate. And these missed ones might show up as the most predominant ones when chickens, earlier fed on this feed, are found to be salmonella positive. And the link is never made, so everyone keeps looking in the wrong place for where the salmonella originally came from. More about this later. So let's look at CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are found in DNA. Now, CRISPR sequences are only found in bacteria. An enzyme known as Cas9 protein that recognises specific DNA repeats is used to cleave CRISPR sequences from the remaining DNA. Now, each serotype of bacteria has its own pattern of short repetitions, and once these are catalogued, serotypes can be identified relatively quickly and easily using CRISPR. One of the benefits this technology brings is the ability to easily find however many serotypes of salmonella they may be in various samples of contaminated feed and or feces or birds, and not only that, but the proportion of these serotypes as well. We've been working with Dr. Nikki Shariat, who is based at the University of Georgia in the USA, and Nikki, along with others, have been looking at using CRISPR to identify salmonella serotypes. Anitox have our own research lab in Georgia, USA, where many studies have been carried out involving identification of salmonella and different methodologies of so doing. we were delighted to support the validation studies of a variant of CRISPR called CRISPR-Seq, and our objectives were to validate CRISPR-Seq by directly comparing it to conventional culturing methods, also to reveal biases that exist at pre-enrichment, enrichment and selective plating steps of conventional Salmonella surveillance, and to investigate different growth rates between Salmonella cerevars commonly found in feed, such as Montevideo and Seftonburg, and those found at lower levels in feed, such as Enteritidis and Typhimurium. So we sent blind samples of feed inoculated with specific serotypes and then compared CRISPR analysis of the feed with the serotype supplied. Furthermore, Anitox sent a series of feed samples which had 2 to 4 different serotypes in equal proportions. The CRISPR technique was then tested to find all the serotypes and in what proportion. These are some of the results of mixed 50-50 serotypes. The left-hand universal pre-enrichment broth bar chart shows the results from analysis of the pre-enrichment broth, and we can see only one result came out at 50-50. It's the same story with the enrichment broth samples. Now what this tells us is that different mixtures of serotypes grow at different speeds in different broths, so we unwittingly get bias in the results depending on which broth is used and which serotypes were there in the first place. Now this shouldn't be a surprise, as in any population of bacteria, some will adapt to the conditions better than others, but it's really interesting to see how different media influence the results and how some serotypes grow evenly when with others, such as Seftonburg and Tennessee together, whereas Tennessee outcompetes Typhimurium even though both started out in a 50-50 mix. This is some other recently published research by Shariat and Fay et al. Over 130 mills sent in around 3 samples of feed each. They found using CRISPR that Infantis was the most common serotype with Heidelberg II. Others included Salmonella redding, Typhimurium and Brenderup, all of which have been linked to Salmonella in humans in the recent past. We know that Salmonella grows in small colonies, which tend to be infrequent and unevenly spread in feed. Now, each of the mills sent in a maximum of 3 samples of feed, so a minuscule sample of each mill's output. The fact that so many samples came back positive for Salmonella indicates Salmonella is present at high levels in these mills, and certainly higher than would be expected from mills in Europe. Similar studies in the EU and the UK by Davies and Wales 2010, for example, tend to focus on dust and spillage from feed from mills, as when Salmonella levels are below detection levels in feed due to insufficient sampling, dust and spillage give a better indication of contamination. Now, not only is Salmonella at high levels, but most of the ones identified are zoonotic. That is, they affect humans and are therefore of particular danger when found in animal feed and so require immediate action. Salmonella become desiccated in feed, which stresses them. Some serotypes recover more quickly and so proliferate faster in pre-enrichment or in enrichment. And we saw variation depending on the type of media used in the earlier slide. Salmonella isolates from carcasses are less stressed as they haven't been dried out, so different serotypes come to predominate when placed in media. And then we wonder why we struggle to find the same Salmonella serotypes in feed as we find in carcasses. It isn't that the same serotypes aren't sometimes present in both. It's just that some serotypes recover quicker in feed and come to predominate in the traditional assay methodology. So we know that zoonotic Salmonella serotypes are indeed present in and spread by animal feed. In light of this knowledge, should we knowingly feed chickens, swine, with feed that has no protection against Salmonella? Surely not. From a corporate responsibility perspective, we should be taking action to decrease or eliminate the risk of chickens or swine coming into contact with Salmonella via feed, and water, rodents, other vermin, contaminated bedding, etc. The paradox is that nearly every integrator and producer will be proactive in tackling nearly all the risks, including water, to which animals have 24/7 access. But how many leave an enormous hole in their biosecurity protocols for Salmonella to enter via feed, which animals likewise have 24/7 access? So all the time and money invested in every other area of biosecurity can be wasted when feed is the forgotten link. So what's at risk? Well, the industry reputation is, including customer confidence and public health, compliance, reputational damage, potential product recall. So how can we solve this? Well, you have three options to make an intervention regarding feed. Only one of those options is effective if you wish to manage Salmonella to the point of consumption by the bird, which is essential when feed is truly a critical control point in your process. One option is heat treatment. It can be highly effective at point of application, but there is zero residual protection against recontamination after treatment. A second option is organic acids. The downsides are they tend to be bacteriostatic, that is, they slow down bacterial growth but don't kill them at anything other than 5 to 10 kilos a tonne application rates. Also, they tend to mask any Salmonella in feed when analysis is done by reducing the pH of the growth media. And, crucially, they offer little or no residual protection. Or you could choose a feed sanitizer, offering high efficacy in controlling both bacteria and viruses, and at low application rates. In Europe, your choices are limited. Finio is the only feed sanitizer on the market. The good news is that there's new independent data to support your investment. This peer-reviewed study by the Animal and Plant Health Agency, known as APHA in the UK, was only published in January 2021. It shows the efficacy of Finio, which is product A, at different application levels. 3 major organic acids were tested at the same time, and 1 kilo a tonne of Finio was shown to be more efficacious than 6 kilos per tonne of any of the organic acids. The other big differentiator over organic acids is the fact that Finio provides residual protection against recontamination and at low application rates, so 1 or 2 kilos per tonne confers very good residual cover, protecting the feed if it comes into contact with sources of recontamination on its way to the farm and to consumption by the birds or pigs. So, back to looking at CRISPR. You'll remember this slide, how we can miss lower frequency serotypes. Work has been going on in the USA looking at CRISPR as a technique for identifying all serotypes in a sample at once. This will remove the chance element we get when we only select 1 or 2 isolates found in a multitude of isolates. So serotypes that grow well in birds and are present but don't tend to show up in feed due to being stressed by the dry conditions will show up using CRISPR. This will allow for much more accurate and quicker tracing of where serotypes are coming from. CRISPR sequences are only found in bacteria, and there are 2 loci in Salmonella. These repeats are from viruses that have infected bacteria and have been incorporated into the DNA of the bacteria and are then passed on when bacteria replicate. So in the future, the industry will have the ability to find all Salmonella serotypes in feed samples or bird samples. Furthermore, it can determine the proportion of different serotypes in a sample. Now, this won't overcome sampling and subsampling errors, but it will help the feed and animal production industries concentrate their Salmonella elimination strategies where they will have the most effect. So these are very positive developments to help further control Salmonella in the feed and food chain in the future. I hope you found that interesting. Now, you're welcome to contact me should you have any questions on scarlton@anitox.com. Thank you for listening.